I'm Dr. Jamie Fong, a Senior Research Fellow at the Department of Physiology in the University of Melbourne.
Hi, I'm Dr. Lin Young Hang. I'm the lead author of this study, which I conducted during my PhD with Dr. Jamie Fong at the University of Melbourne. Our paper, titled Antibiotic Exposure Post-Weaning, disrupts the neurochemistry and function of enteric neurons, mediating colonic motor activity, was recently published in the American Journal of Physiology. Antibiotics are most commonly used to treat bacterial infections in both young and adult populations. In addition to eliminating pathogenic bacteria, antibiotics are also known to devastate the symbiotic microbial communities that are essential for health. This is particularly concerning when antibiotics are used during critical developmental periods, as studies have shown that this can have greater consequences to host physiology and even lead to increased susceptibility to several diseases.
The period during and immediately after weaning, we refer to as the post-weaning period, is an important developmental time triggered by the transition from mother's milk to solid foods and changes in social and other environmental life conditions. Within the gut, the post-weaning period also involves maturation in microbiota, which can modulate gut functions and its underlying neuronal circuitry, known as the enteric nervous system or ENS. But this is poorly understood. In this study, we aim to examine how antibiotic exposure during the post-weaning period affects colonic motility and the neurochemistry and function of enteric neurons.
To do this, 3-week-old mice were treated with either water as our control or the antibiotic Vancomycin, and their colons were examined at 6 weeks of age. We compared the effects of vancomycin exposure post-weaning with an earlier neonatal period to compare the microbiota between these two developmental windows. We found that vancomycin exposure in both postnatal developmental periods significantly reduced the diversity and abundance of fecal microbiota communities. In post-weaning mice, we found a significant increase in fecal microbial communities compared to the neonates, and that vancomycin treatment caused distinct compositional shifts between the two developmental stages. We next examined colonic motor patterns of vancomycin-treated post-weaned mice. These colonic motor patterns are exclusively mediated by enteric neuronal circuitries. We recorded colonic motor patterns of isolated mouse colon using a video camera, and these recordings were then converted to spatial temporal maps for analysis. We found that the speed of long propagating contractions, known as colonic migrating motor complexes, or CMMCs, seen in this video, and also highlighted by these red streaks in the spatial temporal maps, were significantly reduced in vancomycin-treated mice. To investigate whether the slowed colonic motility involved changes in the neurochemistry of enteric neurons, we examined whole mount preparations of enteric plexi using immunofluorescence. In the myenteric plexus, which contains the main circuitry controlling motility, we found that vancomycin treatment specifically reduced the neuronal proportions of neurons that are typically cholinergic, and this is due to their use of the excitatory neurotransmitter acetylcholine.
We next investigated whether vancomycin perturbed the excitability of enteric neurons using calcium imaging. Our mice are genetically encoded to express the fluorescent calcium indicator GCAM3 in all cells of the enteric nervous system. Vancomycin exposure caused more enteric neurons to be activated in response to electrical stimuli. This is shown by these videos from vancomycin-exposed mice on the right, where their myenteric plexus and submucous plexus preparation had more fluorescent cells activated in response to electrical stimulation. The antibiotic had different effects on microbiota and ENS when exposed in different developmental periods. This demonstrates the importance of studying the impact of antibiotics during different developmental windows. Lynn and I would like to acknowledge the grant funding and our collaborators.
To know more about our exciting work, please follow the link to our article.